And it's a great pleasure to be here and to talk about, as Alice said, the topic of splitting and lumping. Or to phrase it a bit differently, when we talk about autism, is there such a thing as autism? Or are there many autisms? Or even, do the different disorders of brain development share more than separates them? And so, to give a preview towards the conclusion, I won't have an answer. I won't be able to tell you that there are 3 autisms or that there's only 1 or there's 10. But I'll talk through the way that we conceptualize the issue and what the data is telling us now about where we are in terms of splitting versus lumping the different disorders and what that might tell us about future avenues to understanding autism as a condition and neurodevelopmental disorders as a set of different conditions. So before I start, a little bit of history. Go back 100 years, and all that there was in what we call now autism was schizophrenia. Over time, as you go into the 1920s and 30s, you began to separate adult schizophrenia from childhood schizophrenia. And this is also the emergence of the attempt to become more rigorous about the classification of disorders in mental health. With the Diagnostic and Statistics Manual, and this the first edition really was the second edition that became publicly used. So this is the Bible of mental health and psychiatry. And it's been iterated over time. So as you go from childhood schizophrenia, around the 1980s, the third edition of the DSM came out. And you began to have infantile autism as a condition, separated from childhood onset schizophrenia, in turn separated from adult schizophrenia. Move forward to the fourth edition of the Diagnostic and Statistics Manual, and infantile autism was split into autism, Asperger's, a term many of you have heard, childhood disintegrative disorders, pediatric developmental disorders not otherwise specified, and Rett syndrome. This again has now been separated from childhood onset schizophrenia and adult schizophrenia. And then just a few years ago, the DSM has been revised yet again into the 5th edition of the DSM, where Rett syndrome has stayed separate, but the autism spectrum disorders have now been grouped together from what was previously the 4 conditions of autism, Asperger's, CDD, and PDD, NOS. So as you see, over the history of the diagnostic categories, we've been lumping and splitting and not quite sure where to draw the boundaries. But of course, some of you might be asking, does it even matter? What does it matter if what we call it as long as we know what it is?
You mean bipolar is not a disease?
There's plenty of other disorders I didn't mention, just in terms of the history of what we would call autism. It used to be called schizophrenia, which was separate from bipolar, etc. So does the name even matter? Does it matter what we call it? And I would argue strongly that it does, on 2 bases. One of them, which I spend most of my time talking about, is in the basis of understanding the biology, helping you understand potential pathways to treatment or understanding outcomes. But even in the absence of that, it can be a simple requirement for clinical, educational, or other services. So there's a good example in some jurisdictions, in some states, if you have a diagnosis of autism, you might be able to have access to different school services than if you have a diagnosis of ADHD. So even though they are just names, they can matter as to how you're able to interact with the services, the clinical or school services around you. It can matter in terms of regulations. It is much easier to get a therapeutic indication for a drug if you're going for a known disorder than if you claim to have a disorder that isn't recognized by the classic manuals. It can make a big difference for building communities. This became really quite noticeable when you went from the DSM-4 to the DSM-5, where Asperger's stopped being referred to as a separate condition. A lot of families, a lot of patients with Asperger's were quite upset at what they considered a part of their identity being taken away. Similarly, if you go to some of the rare disorders, you have these wonderful online, virtual, and real communities that form around people who share the same disorder and want to be able to understand more of what makes this common, what makes this disparate. And then lastly, as you get to more biological understanding of the disorders, and especially when genetics comes in, parents and patients can be quite relieved to understand the origin of the disorder. There's a wonderful quote from a Spectrum News article. Of course, Spectrum News is one of the great contributions of the Simons Foundation to distributing autism knowledge. In this case, they're talking about genetic diagnosis in particular, but I think it holds more generally. Where one parent, for example, used the result to refute a teacher who believed her son was just arrogant. And probably even more poignantly, another regretted having looked for easy solutions and alternative treatments. I would have never considered doing half the stuff I did to the poor child, she said, citing chelation treatments purported to rid the child of toxins. So the term does matter. Within names, within categories, we understand the world around us, and we use services, we build communities, we move towards treatment paths. But then why is it so complicated? Why have we gone from these different terms, from different understandings of whether it's separate from schizophrenia, the same as schizophrenia, whether there's 5 different subtypes of autism, whether Rett's is part of autism or not, whether there's only a single spectrum condition? And I'll start by talking, this will be the main focus of the rest of my next 40 minutes or so, but I'll start by talking about comorbidities. So when you think of autism, there's 2 symptoms that you need. You need to have problems with social communication, and you need to have repetitive behaviors and disorders that affect daily activities. So that's common. Everybody who's diagnosed autism has those. But apart from that, it can be remarkably variable. So about 40% of children diagnosed with autism have average or above average intellectual abilities, and about 25% are nonverbal. So you have the full spectrum from severe intellectual disability, fully fluent to nonverbal, high IQ to low IQ. Somewhere around 15% of children with autism also have concomitant seizures or different forms of epilepsies. About 70% of patients with autism have another psychiatric disorder, with, depending on the study and the sample that you look for, around 30 to 80% will also have attention deficit disorder, 20% that will have OCD or some variant. In this 2009 paper, you can see a table there which lists some of these disorders, where again, any other concomitant mental health diagnosis is sitting at 70%, with anxiety disorders around 40%, depressive disorders fairly low at a few percent, conduct disorders at 30%, et cetera, as you can read down the list. So effectively, you have these 2 shared symptoms in terms of social and repetitive behaviors, but a whole myriad of concomitant disorders that you may or may not share with another child with autism. As I move into the biology of autism, I'm going to focus on 2 aspects of it. I'm going to focus on the genetics, and I'm going to focus on the brain, particular brain structure. And I'll start with a bit of an overview of what we know about the brain structure that might be different between children and adults with autism versus typically developing controls and or patients with other diagnoses. So when we talk about brain structure, we talk about the shape of the brain, overall, the overall volume of the brain, and then local differences, such as is the thick cortex a bit thicker or a bit thinner in the motor cortex? Is the hippocampus a bit larger or a bit smaller? And ultimately, the reason why this could be informative, I would argue, is that we know that there's a relationship between brain structure and function. There's the classic case of a patient who's had the hippocampus removed due to epilepsy surgery who could form no new memories. So these are severe cases. But we also know that as the volumes and sizes and shapes of different structures vary, so does some concomitant behavior. It's very strongly influenced by genes, so it might be a pathway which you can identify how the different genetic disorders associated with autism might act on brain circuits to produce the final symptoms and phenotype. And of course, in addition to genetic influences, there are significant environment influences. But the history of understanding brain structure in autism got off to a bit of a rocky start. And again, just looking at the hippocampus as an example, if you go through the literature from 15, 20 years ago, there was some clear evidence from 2002 that the amygdala and the hippocampus, so 2 structures in the deeper part of the brain, are enlarged in children with autism, or are significantly reduced, or if you like, neither of those are not different. And this tells us a bit both about the state of research at that point in time, where most of the samples were fairly small. It might be 20 patients versus 20 controls, or in those scopes, which we know now, because there's significant heterogeneity in the disorder, is insufficient to capture all of what's autism, which in and of itself is informative. If you have 20 patients with Huntington's versus 20 controls, you will get a pretty accurate picture of what happens in the brain in Huntington's disease. You will not with autism. You'll get these kind of disparate results. So you have to start moving towards larger samples in order to really be able to understand what's happening. So this is a recent study which gives you a bit more of an overview of where we're standing now, or in 2019, which combined multiple data sets looking across almost all samples that had been collected and ending up with a sample size of around 2,000 children with autism, 2,000 controls or thereabouts. And looking at the effect sizes, displayed here on the y-axis, estimated in terms of standard deviations, or Cohen's d. So effectively, if you're 1 standard deviation away, then you're around 70% of the population is going to be smaller than you. So when we're talking about effect sizes of 0.1, 0.2, we're talking about fairly small effect sizes. They're not easily going to separate patients from controls. But given sufficient sample, can still separate out effects that exist across the whole population. So going across this table, you can see that almost all of the effect sizes are positive, meaning that the CV, or cerebral volume, is enlarged in patients with autism. which is especially reflected in the mean thickness of the cortex, or mean CT there, somewhat less so in terms of TBV, or total brain volume, and also somewhat less in terms of total subcortical surface area and white matter volume. And looking across the individual studies in these samples, you can see that, again, any individual study might or might not encompass zero, or might or might not be statistically significant. But in aggregate, once you sum across all of these studies, you begin to see that there is a subtle but significant increase in the thickness of the cortex in autism versus controls. This is spatially localized. So the areas you see in red here in the top part of these images, see my mouse, are areas that are significantly associated with autism compared to the areas in white, which are not significantly associated with autism. So you have these foci in the temporal lobes, as well as in the frontal cortex in particular. So picking one of those out, in this case in the left superior temporal gyrus, indicated with asterisks in that figure, you can again see in any one individual study sample, which are the different columns on the different rows in this table. In some cases it's significant, in some cases it's not, but in aggregate, summing across the whole 2,000 subjects in multiple different samples, you can see that there is an effect. Using just our sample from Toronto, I'll give you again a bit of a different overview of what this looks like, where I'm showing with confidence intervals across different brain areas, subtle increases in the inferior temporal gyrus, increases in the superior temporal gyrus again. So in this case, blue is patients with autism, red is controls. Not much change around the motor areas. And then decreases in volume as you go subcortically. So in this case, in the putamen, in the thalamus, and in the hippocampal formation. So it's a bit of a whirlwind.
Is this evoked or spontaneous activity?
This isn't activity, this is size. So it's just the volume of the hippocampus, the volume of that structure. So in that sense, it's environmentally influenced when you look at brain structure, but it's really mostly driven in development. So in that sense, you're seeing the cumulative impact of genes plus environment up until that age, and it doesn't fluctuate much day in, day out. But again, the take-home message from this is, given large enough samples, you can identify differences between patients and controls in brain structures. But if you look at the individual data points, as I'm showing here, you can very clearly see that this would not be diagnostic. If I look at the blue points, and this is the surface area of the left temporal pole as one example, versus the red points, there's a giant amount of overlap. And if I were to try to diagnose based on this alone, my accuracy would be barely above chance. Also showing the same thing against age on the graph on the left, on the right. You can see that again, there are significant patterns that separate across age, but there is no easy way to separate groups other than in large numbers using statistical inference methods. So can we get a bit more information by letting the data tell us if there's subgroupings within these that actually drive the data in different directions? So this is a paper from a McGill group led by Boris Bernhardt, along with the group here that has created the ABIDE data set for large population samples with Adriana Di Martino, where they used clustering methods. So this is, again, a data-driven technique where you put in all of the input samples in terms of thickness, brain structure, brain volume, size of certain brain areas, and then you cluster it together to see which subjects and which brain areas associate with each other. So in this case, each individual color point in this complex diagram there corresponds to one brain area for one subject. And if they did this, and they found that there are 3 different subgroups that emerged within their autism data set. And if you look at the individual images from these 3 groups that they labeled very creatively, ASD1, ASD2, ASD3, you can see that in one group you have these increases in cortical thickness shown in red, pretty much distributed widely across the cortex. In another subgroup, ASD2, you had decreases mostly in the posterior parts of the cortex. And in the third, the brain wasn't affected much at all. And what was interesting, when you then try to take this information and say, to what extent can I predict outcome or the amount of severity or phenotype of the patients given brain structure, whether I cluster the subjects into these 3 groups or whether I just do ASD versus control as one overall measurement, what they found is that the ability to predict the ADOS score, so one of the common instruments that you use to understand overall phenotype severity in autism, was fairly poor, non-existent, if you just did an overall prediction of the ADOS score using the whole sample. But once you use the subgrouping into 3 different anatomy-driven subgroups, the prediction actually became pretty decent. So again, it's beginning to tell us that overall, if you treat autism as one collective whole, in terms of at least the brain structure subtype of it, you're not getting very clean answer of what the disease represents at the biology end of it. If you begin to subdivide it into, in this case, 3 subgroups, you can begin to predict outcome to a somewhat better extent. So if I go back to my diagram, maybe what we're going to now is from 4 labeled autisms in DSM-4 to the autism spectrum in DSM-5, maybe subgrouped into these 3 different anatomy-driven subconditions, the ASD 1, 2, and 3. So switching a bit over to genetics, what kind of information can we glean about the lumping or splitting of autism and related conditions? So first, if we're talking about genetics and autism, we're talking about something that's quite distinct from many of the other mental health disorders. So for those of you who've looked into Alzheimer's as one example, what we know there is that there's some common variants, such as the ApoE gene, where if you have a certain copy of that variant, your risk increases significantly. If you don't, the risk doesn't. There's really nothing like that that's yet been identified in autism. We don't have any of these common variants that are shared in the population which give you a prediction of increased risk or not. Instead, what we have are these really significant number of rare variants or genes that are not very common in the population at all. But if you have them, they're highly penetrant, which means your risk increases significantly. So from a recent paper in 2017 from the Steve Shearer's group in Toronto, they came up with a list of a large number of genes, and they're all listed with poetic names like CHD8 and ARID1B and other things that just roll off the tongue. But ultimately, the story that comes out is that it's highly heritable, and heritable indices, again, depending on the paper that you study, go up to around 0.8 or 80% heritability. But there are hundreds of identified genes, with no single gene accounting for more than 1 to 2% of the cases. And usually, if you go for a larger sample, in around 20 to 30% of that cohort of patients, will you be able to identify a likely putative genetic cause. And in the other 70 to 80%, you will not be able to identify genetic origin. Doesn't mean that there isn't a genetic origin, and given the heritability, it's almost certain that there is, but we've not yet been able to find it. So given these large numbers of genes, each accounting for a few cases only, maybe the way we should think about this kind of clustering is that you go from the subtypes of autism into autism spectrum disorder, into a large variety of individual autisms based on the genetic origin. So maybe there's a CHD8 autism, maybe there's an ARID1B autism, maybe there's a DYRK1A autism, and so on and so on. To a large extent, this would give us very biologically driven information about the subtypes, saying here's a gene, we know what that gene does, or we have some idea of what it does, and that causes autism in a particular way in this one subject. This might be the case, but if it is, we're also in a bit of trouble as a community, because if you look at most of these genes, you will find 5 to 10 to 15 patients around the world who carry that mutation. And so the ability to then synthesize and be able to think of treatment paths or other interventions is almost impossible. It's a very long road ahead. Also, in that case, you're still missing 70 to 80% of the cases where we do not know what the genetic cause is, if there is any at all. So one way you can begin to think about this is by, rather than looking at the individual genes on their own, is looking at gene groupings. So in some ways you can say that we know what gene does to some extent, and we know which other genes it interacts with. So it's almost a bit like if you had apple, pear, orange, rose, and orchid. You could say that the apple, pear, and orange correspond to fruit, the rose and orchid to flowers, each of which are plants, and maybe you can make inference on fruit versus plant as opposed to on the individual gene itself. And there have been multiple attempts to do this, and this is again just one example, where looking at many of these individual genes and seeing how the genes interact with each other, you can come up with some clusterings of these genes themselves. And really the 3 that have been consistently identified, and again citing a paper from 2017 here, are genes that deal with transcriptional regulation, synaptic and neuronal adhesion, as well as RNA processing. So I'll back up just a little bit to talk through that. So when we talk about synaptic and neural adhesion, we're really talking about the way that cells communicate with each other. So to give a very simplistic textbook drawing of a neuron, you have a neuron sitting here at the top, and it sends its axon out, which is where signals are then propagated down to communicate with another neuron. And the way that this then works is you have these neurotransmitters that are released that are then taken up by the neuron on the receiving end, and that's how signals propagate. Now, in this very complex picture, you can see that there's a lot of molecular pathways that are involved in making this work. And the ones that come up fairly consistently are these adhesion genes, or the things that span from the postsynaptic to the presynaptic neuron. So effectively, you can think of the closer anchored the neuron that sends the signal, neuron that receives the signal are, the better the communication. And you can regulate the degree of effectiveness of communication by anchoring less well or stronger. And so if you influence these genes, then you're going to get slightly disturbed neuronal signaling, which maybe at the end of the day then results in some of the symptoms that we see. So that was the top component here, the synapse and neuronal adhesion components. Transcriptional regulation is getting more at the way that DNA itself is processed. So again, very simplistic high school biology. You really go from having your DNA that's then transcribed into RNA, which in turn is translated into protein. So that's kind of the pathway that you go from having the code in the DNA to the things that actually do the work. And along that way, there's multiple things that can both interfere and that are necessary to make it work. So when we talk about transcriptional regulation, is the ways to either enhance or activate transcription, so turn DNA into RNA, or that can inhibit or repress transcription. And so again, when you're influencing activation or repression of transcription, you're not necessarily influencing one gene, you're influencing hundreds of genes that in turn are then either better or worse repressed or transcribed, and therefore might change subtly how the DNA goes to protein. And that's the key to most of these symptoms, is you're looking at autism-related neurodevelopmental disorders, which by very definition are something that's not severe because you're surviving. And so you're not going to have genes that completely disable brain signaling, because if those genes exist, and they tend to result in non-viable embryos. On the other hand, you have genes that are viable, you have perfectly functioning individuals, but there's subtle perturbations in signaling or in transcription that then have these outcomes in terms of the social behaviors, repetitive syndromes, and potential epilepsy, attention problems, et cetera, down the road. So then the third of the group is RNA processing, which is very much related to transcriptional regulation, where again, as you go from DNA to RNA, you then take the RNA transcript and turn it into protein. And then again, there's multiple things that the body does, the cell does, and when it goes from RNA to protein in terms of splicing the RNA, et cetera, in order to result in different types of proteins, where again, if you're subtly changing that, you're having subtly disturbed outcomes at the end of the day. So if we look at these elements, maybe the way we should be thinking about autism as a spectrum disorder that's influenced by many genes, but these genes in turn are linked to certain pathways. So you have genes that are acting on transcription, genes that are acting on synapse adhesion, genes that are acting on translation, and they in turn might share some common phenotype. But back to the comorbidities. So we're talking about autism as its own thing so far and finding ways of looking at genetics or brain structure to subdivide it. But are we really confident that we should be looking at subdividing within autism? So again, looking at these comorbidities here, you can see that a high proportion of children with autism also have ADHD. Or the other way around, high proportion of children with ADHD also have autism. And then there's seizures, there's anxiety disorders, et cetera. So maybe one way to look at this is to ask, are we actually separating out ADHD from autism when we start looking at the biological substrates? Or are we talking about a continuum where we actually have more shared than disparate things? And some evidence from that, again, comes from genetics to start, where there's a wonderful study from Sweden, and the Scandinavians have these brilliant health records, which let you link health outcomes to other samples in ways that are very difficult to do in the North American system. And what they asked is not, is autism heritable? We know that at this point. But does having a diagnosis of autism predict having a diagnosis of ADHD given relatedness as a measure? And the answer, it does to a remarkable extent in a diagram that clearly suggests heritability. So to walk you through this, again, this is the odds ratio, or the likelihood of having the one disorder given the other. And so if you look here in monozygotic twins, if you have a monozygotic twin with autism, there's a 16-fold increase in likelihood of having the other monozygotic twin have ADHD. As you go to dizygotic twins, so now you're not sharing the exact same DNA copies anymore, that goes down to an odds ratio of around 4. Full siblings are the same as dizygotic twins, which makes sense. Half siblings, odds ratio goes down to 2. Parental half siblings, around 1, half cousins 1.05. So this classic decrease that the closer the genetics are, the higher the risk of having the other disorder given one of the disorders. So it's clearly indicating there's something significantly shared about them. And maybe, and we don't know the process yet, there might be subtle pathways during development, subtle environmental influences, or influences of other genes that move you from having something slightly wrong with the way that your DNA goes to protein, the way that the synapses signal with each other, that then pushes you to having more attentional symptoms or more social symptoms for reasons we don't understand, but that still derive from the same underlying basis, given this large heritability. Sorry, were you?
Yeah, but considering that these are concordant genetically for the monozygotic twins, 16% is actually quite low.
16 times, not 16%.
16 times. So are you looking at the concordant twins that don't display the comorbidity in terms of examining protective factors, either neurochemically or genetically? So the twins that are
Right. So the question for those who couldn't hear is, can we look at the other way around and say, if you're not sharing it, to what extent does that give you something about protective factors? So that's a fascinating question. And so first, this wasn't our study. This was something from the literature. And I don't know if they have. It's certainly an important question that is being interrogated and probably should do more of, is not just what provides risk, but what provides lack of risk, or what provides some kind of a way of giving you an out on the disorders. So I'm going to go to another example, which is now if you actually share the same genetic load, so that we identified the genes, how do you begin to dissociate the disorders? The example I'll use is the sex chromosome aneuploidies. So I'm sure most of you know about Down syndrome, which is an extra copy of chromosome 21. The other common type of chromosomal duplication or deletion like that tend to occur on the sex chromosomes, so the X and the Y chromosome, where you get, for example, Klinefelter syndrome, which is 2 X copies of X, 1 copy of Y. And you get other, even more rare disorders where you have 3 copies of X, 4 copies of the X chromosome, or 4 X's and 1 Y, and so on. So collectively, these are termed as the sex chromosome aneuploidies. And most of these individuals are, again, fairly high functioning, but tend to have some mental health association with the genetic load that they have. But then if we ask what that is, we again see a variety of the diagnoses crop up. So for example, in this case, I'm comparing XXX, XXY, and XYY, so single additional sex chromosome. And if you look at XXY, about 27 of these 51 kids also have autism. Sorry, 21% have autism, 63% have ADHD, 24% of mood disorders, 6% of anxiety disorders. If you go at XYY, 14% of autism, 66% of ADHD, and you can go down the rest of this table as you read through it. So the same genetic lesion, the same genetic event, causes differential diagnosis in terms of autism, ADHD, etc. But the diagnosis, having a mental health diagnosis, is significantly more likely if you have these duplication of an extra chromosome on X or Y. Going back to the brain imaging studies, we have a large cohort in Toronto where we're looking at not just autism versus typically developing controls, but also comparing that to ADHD and OCD, as well as some of the more rare disorders, and trying to ask, to what extent are we actually separating out ADHD from OCD from autism? And in the first study that we published on this in 2016, we looked at differences in white matter, so the different connections between brain areas. And we found that the most robust finding was actually all of the neurodevelopmental disorders, so in this case, ADHD, autism, and obsessive compulsive disorder, separating from typically developing controls. And then looking within those groupings, we could see differences, and anything in color you see in the white matter is a difference, and anything not in color is no difference here. We could identify differences between our autism patients and our control subjects. We could identify differences between autism and obsessive compulsive disorder, between ADHD and the typically developing children, and between ADHD and obsessive compulsive disorder, but we could find no statistically significant differences at all between ADHD and autism. Looking at networks in the brain, so rather than just looking at the structure or the direct connectivity between them, we can ask to what extent do different brain areas signal together. So as one area is active, is another part of the brain active as well, using functional MRI. We did this in the same population, so again, kids with autism, ADHD, and OCD versus normally developed controls. And what we found is that across a series of these networks, that there were indeed differences between our patients versus our controls, but we could not actually separate out the patient groups at all. So we could find that if you have a mental health disorder, you will have somewhat differential signaling, but we couldn't separate ADHD from OCD, we couldn't separate ASD from ADHD. So, as I said here, some network increases, some decreases, but with neurodevelopmental disorders as a whole, not with the subdivisions between them. So maybe we've been looking at it wrong again and trying to subdivide the different autisms or trying to decide whether autism should be split or merged. Maybe we have to start incorporating the other disorders as well. Maybe we don't have a clean separation between ADHD, OCD, and autism the way that we clinically thought we would. And again, to take a step back, the diagnoses are fairly reliable. So it's not that I'm saying that a clinician can't separate out ADHD from autism reliable from one clinician to the next. There's some debate about whether there are some boundaries where that isn't the case. But for the most part, it's indeed, the diagnoses might be dependent on a few symptoms, but the fact that we have this attentional domain as one set of symptoms and social as the key thing that separates the different patients from different diagnoses might not actually reflect underlying biology and the complexity and the heterogeneity of these disorders themselves. So going back to clustering, or then using the data to drive patterns in terms of groupings, what if we use the richness of the data set that we have, where we have the same scans on kids with ASD, ADHD, and OCD? Do our clusters actually begin to pull out the different diagnoses or not? So we did this initially using, again, brain structure as our measure, and we identified 10 separate clusters within that. And so each of these 10 clusters are on the x-axis, with the percent participants in each cluster in the bars, and the different colors corresponding to their primary diagnosis. So the dark blues or purples are ASD, blues are ADHD, greens are obsessive compulsive disorder, and yellows are the typically developing controls. And the first thing that you can tell is that there isn't a single grouping in this data-driven analysis that is purely one diagnosis. In each case, we're mixing in some kids with OCD, some kids with ADHD, and often even merging with the controls. You do have some separation that the first few clusters have controls in decreasing level, and the last few have no controls in them at all. But still, it's not coming out that if you do a data-driven analysis, you're not coming out with the initial diagnoses that you put in. Instead, you're coming out with mixes of the initial diagnoses. Similarly, if you use this functional signaling to what extent do the signaling between brain areas couple, and again, try to do some data-driven clustering on that, we again, in this case, come up with 2 clusters, given this particular measurement. And if we look at the proportion of participants in each cluster, we have a cluster 2 in this case, where most of the typically developing controls go. But again, there isn't anything resembling a clean separation of the diagnostic groups. You have some ability to separate OCD, which looks somewhat similar to controls in these cases. But some of the ASD patients go there, some of the ASD patients go in the other group, etc. So using data-driven techniques, and of course, these can always be challenged by if you have other measures, you might get other clusters coming out. But using at least these measures of brain connectivity in terms of white matter imaging measures, brain structure, and brain functional coupling, we're not finding the initial diagnoses again if we let the data tell us what the groupings should be. But we are finding subgroupings within that. So maybe we should be merging the different neurodevelopmental disorders and then subdividing them again, depending on how you end up doing the clustering, into 2 different types of neurodevelopmental disorders, but that do not cleanly reflect our initial diagnoses, or maybe 10 different types of neurodevelopmental disorders, etc. So how do we merge this picture, where we have the neurodevelopmental disorders really sharing lots of features with some subgroupings within that, with the genetic information that we talked about earlier, with these individual genes sitting in certain pathways that might act in terms of understanding about what's happening, what was the initial mutation that might cause the symptoms and the phenotype that we're seeing. So again, as a reminder, we're sitting in this game that we have rare variants in autism that are highly penetrant. And what this means in terms of the research context, that it's very difficult to study the individual cases associated with a single gene because they're so rare. So for even the most common mutations like CHD and ARID1B, you're looking at the low 10s to 20s of numbers of patients, even if you aggregate across all of North America, never mind just one local site. There are some copy number variants where you can do this kind of thing, and the most the classic example of that is the 16p11 copy number variance, one region on chromosome 16, where both deletions and duplications have been associated with autism as well as some other related disorders. And there's been some wonderful imaging studies, including the ones done by the Simons Foundation here, which have shown these interesting focal patterns of certain areas of the brain that are likely affected if you have this particular mutation, and that work in a dose-dependent way, where a deletion gives you the opposite result of a duplication of this region. But again, it's very challenging to do this for all of the list of mutations because of the rarity of the mutations. But this is where we start thinking about using mouse models and where they come to the fore as one of their strengths. And so when you're looking at the mouse, we get about 85% or so identical genes between mouse and human. So we can start to ask, is there a version of a mouse of CHD8 or ARID1B, and then can we modify it and see what happens? And of course, in mice, we can then breed the numbers that we need to get adequately powered samples. And there's the wide array of genetic tools developed by the scientific community that have really found ways to modify the genome in very selective, very targeted, very precise ways, as well as some tests for social and repetitive behaviors. And we've developed some comparable MRI methods to really give you the same signal when we look at mouse versus human. So the basic idea is that you create your mutant mouse by inducing a deletion or a duplication of a certain region or a certain gene. You then breed it with a wild-type mouse, which gives you some mice that carry the mutation, some mice that do not, that you can then begin to compare to each other after you have sufficient numbers. And then we've added these mouse brain imaging methods on, which you can then drive with the same kind of analyses I was showing for the human data to ask, what does this particular mutation do now in the mouse context with everything else being held steady? So every other gene is the same, except for the one that you've mutated. We then developed these ways that Alice mentioned at the beginning of imaging multiple of these brains at once. So you get these fairly high resolution scans where you're not seeing individual cells, we're not at that level, but we're seeing the regions in ways that are comparable to what you would on a human MRI to begin to ask, is the size or the shape or the volume or the connectivity between brain regions affected given the mutation? So we take this data, and to focus on brain structure, for example, we might have an atlas where every color corresponds to a different brain area, which we can backpropagate with individual scans to then give us volumetric measurements. And we can do this in multiple different mutations. So to show one example mouse result, I'm looking at 3 individual genes, each of them associated with a high degree of confidence in autism. So there's CHD8 at the top, and the red colors indicate increases in volume, blue colors indicate decreases in volume. And in this case, I'm going from the back of the mouse brain, so you have the cerebellum over here, and then different slices towards the front of the mouse brain, or the olfactory areas of the mouse brain. And you can see that most of the cortex and some of the amygdala is increasing in volume if you have deleted a copy of CHD8. If you've created a single point mutation in neuroligin 3, a gene that's associated with synapse adhesion, then you begin to see significant decreases in volume across most of the brain. And if you've deleted 2 copies of Nerexin 1 alpha, you have a focal area of increases in volume, again, somewhat different. So the first simple message by looking at this is here are 3 mutations, each associated with autism, so the construct validity is high. We know that this particular gene has a high degree of effect in humans, showing very different extensive effects across the mouse. And as a small aside, in those cases where we can have MRI studies in both humans and mice, we tend to find that there's a decently reliable effect that if you see a certain area of the brain, for example, in 16p11, decreased in size in humans, it tends to be that same area that's also decreased in size in mice when you're talking about the exact same genomic modification. But we can look at that a bit more closely if we're talking about mouse to human again. When you don't have the individual mutation, the way, for example, you're talking about 16p11 in humans, but instead talking about idiopathic autism or simply the diagnosis. And we did this initially fairly simplistically, with further work to be done to do this in a more clever way, where we simply took the brain areas where, due to what we know about the evolution of the brain, we know are conserved. For example, you can say that the dentate gyrus of the hippocampus maps to the dentate gyrus of the hippocampus. The striatum maps to the caudoputamen. And then there's multiple other brain areas where the association is less well understood, so especially some of the frontal cortical areas, which for the sake of argument, just for now, we've decided to ignore. So if you then look at the match between to what extent does a single mouse mutation tell you about autism as a whole, it's actually quite poor. So in this graph here, what I'm showing is the mouse model on the x-axis, in this case the mouse model for neuroligin 3, where every dot corresponds to a brain area, and then the unit that I'm measuring in is effect size. And on the y-axis, you have our idiopathic autism population, and again, the same corresponding brain areas. So if I look at the correlation between what's affected in the mouse versus what's affected in our idiopathic autism case, you can see the correlation if neuroligin 3 is the mutant that you have is fairly poor. And if I look at a different one, so one of the other examples I showed earlier, neurexin 1 alpha, you're again seeing that there isn't that much association. So the mouse doesn't predict the human data very well. Or another way to think about it is the single mutation doesn't predict the idiopathic case particularly well. So what might be happening is instead is that when we're looking at the idiopathic case in the clinic, the individual mutations that contribute to it aren't all contributing to it in the same way. Instead, you're seeing something which is an aggregate of multiple individual mutations. So we can also test that in the mouse, and we've been trying to do so as much as we can. It's an ongoing effort, where so far we've looked at a total of 125 different mouse models of individual genes associated with autism through human genetics. And then in each case, we would have around 10 mutants, 10 controls, and look at the effect size of what's happening in the brain in each of these individual mutations. And this really work has been led by Jacob Elligood from our lab, who's still in Toronto, still working on this, still trying to increase our numbers and get more insight into what this tells us about the disorder. So once we have these 125 different mutations, we can ask, across all of these different mutations, what are some of the most affected brain areas, which is what I'm showing here, where you can see that areas of the striatum, areas of the hypothalamus tend to come up as more likely affected than some cortical areas, for example. But we can then take that back to the human and ask, can we do a better job of predicting or of associating the human idiopathic case to this large now population of mouse models than any individual mouse model can? And the answer is that yes. It's of course not a perfect association, which is to be expected because you're still talking about a small furry rodent with a tail versus a human population. But it's a clearly stronger association where areas that tend to be decreased in size in humans, such as the striatum or the dentate gyrus, tend to also be decreased in size in aggregate across these multiple individual mutations once they're grouped together as our case of modeling autism as a collection of rare disorders. So given that we have this large population of mouse models with sufficient power to identify what's happening with each individual gene, we can start to ask some questions about the origin of that phenotype. So when you're thinking about a reduction in volume, so let's say that the striatum is smaller in idiopathic autism, it's small in this collection of mouse models, there's really two things that might be driving this kind of decrease in volume. There's the classic case where most models or most patients have a slightly smaller striatum, and that gives you the effect size that you see. Or there's the alternate, where the mode of the distributions might be the same, so most patients are unaffected, but there's a few that have a significantly large reduction in the volume of the striatum, or whatever structure that you're looking for. Or there's a few mouse models that have a significantly reduced volume of the striatum, whereas most aren't actually affected. And it seems to be more the latter that we're seeing. So in this case, here's a subset of these mouse models where every bar on the x-axis is an individual mutation. And on the y-axis, we have the effect size now for overall brain volume. And it's clear that you get a wide gamut of significant increases in volume over here. For example, a mutation associated with PTEN, which we know is a macrocephaly gene, results in significantly larger brains, also results in significantly larger mouse brains. And on the other axis, you have MECP2, or the gene associated with Rett syndrome. We know results in smaller brains in human, results in smaller brains in mice. And you have everything in between. If we look at brain volume overall in humans, you have a small increase in brain volume. And if we look at it in mice, overall, you also have a small increase in brain volume. But if you look at the distributions, what you'd expect under the null effect, so if all mice were just randomly distributed, is what's shown here in the reddish colors. And in blue, it's the real data that we're seeing. Where again, what you're seeing is this wider tail, where there's some mutations resulting in significant increases in brain volume, PTEN being the classic example, others resulting in significant decreases in volume, with most showing no effect at all. So plotting some of these individual points gives you that same message here in terms of looking at the large effects that you see, but only in a subset of the models. If I look at the striatum, and now I'm looking at the same kind of plot in humans and in mice, where the mutants are in, the wild types are in red, the mutants are in blue. You can see that the means indicated in the dashed lines are slightly lower in terms of volume in both the mouse and the human populations, but that the modes of the distributions are really quite overlapped. And instead, what you see is a wider extent on both the positive and the negative side for both the mouse and the human. Sorry. Which drives the means decreasing a bit, and that the distribution is slightly shifted, whereas the mode remains the same. What's interesting about that is if I take different brain areas, it ends up being different mutations that are driving these decreases depending on the brain area that you look at. So if I look at the decreases in the corticobutamen or the striatum, it's really driven in this case by integrin beta 3, FOXP1, neuroligin 3. And if I look at the decreases in a part of the cerebellum, it's driven by CYFIP1 and GRAIL2. So arguing that what we're seeing, we're looking at the idiopathic cases, is a strange mix of different regions of the brain being influenced by different genes in different subjects. We can go back to the idea of then clustering and asking if we let the data tell us about how the population subdivides, what comes out if we look at the mouse? And given that we're not that confident in saying that there's definitely 3 clusters or 4 clusters or 10 clusters, I'm showing this across a number of subdividing the mouse population in a data-driven way based on the brain structure alone, going from 2 clusters to 10 clusters. And what you can see is if you divide it into 2 clusters, you don't see much of an effect across the brain. As you start increasing the number of clusters, you start seeing, for example, cluster number 1 having some increases in volume in the medulla and brainstem, and so on. As I go further down, you start seeing more specific patterns come out as you let the data subdivide further and further. As you get to cluster 7, you start getting some quite significant reductions in some clusters in terms of volume and increases in volume in other clusters, etc., as you go down to 10 clusters. You can now start to ask associated questions about this, such as, are some of these clusters the more pure autism genes, some of them the more syndromic genes? Are they sitting on different signaling pathways? And for that, we went back to, again, the wonderful database created here. This is now the slightly outdated version of dividing the genes into different levels of confidence, 5 different levels of confidence, or syndromic genes. And then this allows us to ask that given these genes, again, are we getting a pure autism cluster being the high confidence genes, and the rest of the more other neurodevelopmental conditions, or not? So looking at it in this graph, it's the same clustering based on anatomy that I showed before, where each individual line has a color between 1 and 5, with 1 being, and the red colors being the high confidence genes, and the more beige colors being the low confidence genes, and the whites were the unannotated ones. And then the mean colors for each cluster is reflected in the bars. So the key take-home message in this spaghetti graph is that you're not getting a clean dissociation between all the high confidence genes doing one thing, and then the more syndromic other associations doing something else. We can ask that explicitly, do the syndromic mutations, so this would be for things like Rett syndrome, Fragile X, Sanfilippo syndrome, et cetera, do they cluster together differently from the genes that are not associated with these distinct syndromes? And again, the answer is no.
What was the y-axis on the previous?
It's the same y-axis, it's number of models. So it's 125, each line corresponds to an individual mouse model. And then clustering, and as they move from cluster to cluster, it's where the lines go in this kind of Sankey diagram. But again, the take-home message from these is that you're seeing, what we're seeing in the human cases, a mix of different genes associated sometimes with syndrome, sometimes with clean autism as such, or in terms of the effect on the brain, grouping together not by subdividing clean autism from syndromic autism, but intermingled instead. And if we look at the signaling pathways associated with these genes, you're getting a lot of genetics acronyms. But again, what seems to be coming out is that there's a few of these subpatterns that are coming out with signaling pathways, such as synapse adhesion, the MAPK pathway, the Wnt signaling pathway, et cetera, which somebody wants to talk about later as to what they are. Happy to, but I won't go into detail now. Which seem to begin to actually separate out these groups a bit. It's not what the individual gene is, not its high confidence association with autism, but more what signaling pathways that it sits in. So to sum this up, maybe what we're doing is taking these individual neurodevelopmental disorders, which are coming from a mix of primary diagnoses of autism, ADHD, OCD, and others, being able to group them using the data that we have into different subtypes of neurodevelopmental disorders and associate them with different signaling pathways. So I'll sum up some conclusions and then 2 or 3 more slides on future directions and one example, where effectively the argument I hope to have made today is that there are signatures of autism in the brain, but they're really not pure. There's no clean, very strong driven biological signal. though there is something once you have sufficiently large samples. No one mutation models all of autism. So a CHD8 mutation results in a different effect on the brain as an ARID1B mutation as a 16p11 copy number variant. The different signatures we see when we look at all of autism versus typically developing controls, the different subparts of that are being driven by different mutations. So it's not just that there's some kids who have a smaller cerebellum and a smaller striatum, or more signaling in the white matter versus more connectivity in two brain areas. It seems to be different kids, different mutations driving these individual differences in the larger population. And both the human and the mouse data cleanly suggests that there's more than one autism, but there's not one identified per gene. It's mixed in with the other neurodevelopmental disorders, especially with ADHD. And then hopefully, as we're starting to really understand what these subgroupings might be or what some cleaner subgroupings are, maybe they lead to better paths to treatment. So in terms of going forward with this, probably the biggest drawback from almost everything I talked about is we're talking about either older children in the human studies going from age 5 onwards or adult mice. And so a big way we want to go is to go more towards imaging development. So we have the methods to really image a mouse starting at postnatal day 1, even going in utero if we have to. And we can really see the brain unfold in this case as we do that. And there can be some striking findings if you look across development. So I'll just give a single example from ARID1B, it's one of the more high confidence associations in autism, where we've imaged across development here. And in these busy slides, what you're seeing, if I have my mouse here, is the effect at 4 days of age, 6 days of age, 8 days of age in males, and over here in females, where you can see that some patterns in the brain are clearly present throughout development, whereas some others begin to emerge over development. And there's some fascinating sex differences that begin to come out as well, where if you look at the final time point, so at an adult time point, the male and the female ARID1B mutation carriers look the same. But the pathway of how they get there is quite different. So if you look at the first time point in the cerebellum, for example, you see increases in volume in the female mice and decreases in volume in the male mice. So in this case, if you actually look at it as an analysis of variance, we have a strong genotype effect, but we also have a sex by genotype interaction, an age by genotype interaction, and a sex by age by genotype interaction, meaning that if we're just looking at adults and just looking at males, we're still missing a significant part of the picture. And just to illustrate this in graph form, what really seems to be happening here is that the phenotype is present very early in the male mice. It's the same in the female mice adulthood, but it emerges later, a somewhat different developmental trajectory. Just one example, we clearly have to doing this in more different types of models, but we have to really bring back developmental trajectories and sex difference as well when we want to start figuring out subtypes and subgroupings. So with that, I'll end. I thank the many people I've worked with directly in Oxford and Toronto, and funding sources, including the Simons Foundation. Very grateful for your support. And then there have been many people around the world who've contributed to this type of effort, which didn't all fit into one slide, so we have it as a little movie here of those who helped us. And thank you for your attention.